Afternoon.
So, very good. Today we have a lecture about Herpesviruses family, and it will be our first part of today's lecture. And in the second part, I'll give you an overview in microbiology and some case immunology course to refresh your memory about what we studied. And it's our last lecture total over the semester and in the year of our meetings, I would say. Okay, so as I said, first of all, I will give you a part about Herpesviruses family, and also I will tell you a few words about prion infections, so unconventional viral agents also. During the lecture, please keep your microphone off, and between the parts, so in the middle of the lecture, please write down in the main chat your name and group number, so I will understand who are present and absent during the lecture. So please do it in the middle of the lecture. Okay, so I will start. And first of all, of course, I will start with Herpesviruses family, and I will tell you about just a couple of herpes viruses. So all herpes viruses are can be divided into mainly 8 members, which include first and second herpes simplex virus, which cause of fever, blisters, and genital infections. One of that you saw on the first primary slide. Third type, it's a varicella zoster virus, causes chickenpox and shingles. Also, cytomegalovirus, which infect the salivary glands and other viscera. Epstein-Barr virus associated with infection of the lymphoid tissue. Human herpes virus 6 and 7, which cause roseola. And the last one, herpes virus 8, which very associated with sarcoma Kaposi syndrome. Kaposi sarcoma, very common clinical manifestation, and I would say, yeah, okay, it's a particular disease during AIDS, the last stage of HIV. First of all, let me tell you about herpes simplex virus first type, or herpes virus 1, HSV1, as we can decide. What's the main characteristic of this virus? It's a DNA virus, which has double-stranded linear genome. It's enveloped virus, but capsid has icosahedral symmetry. In this characteristic fit for both for herpes simplex 1 and for herpes simplex 2 also. It's the member of simplex virus genus. Once again, how it looks like. Icosahedral capsid, which include double-stranded DNA, surrounded by glycoproteins, which we call supercapsid, and only virus that obtain enveloped by budding from nuclear membrane. So as you remember, viruses with the envelope with the supercapsid usually take a part of the membrane during replication cycle. Primary enveloped viruses take a cytoplasmic membrane on its own surface, but this virus took nuclear membrane on the surface. So Although internuclear inclusions body from infected cells because virus replicate in nucleus, and it's a cytolytic virus, so destroy the cell. Typically, herpes simplex 1 infect above the waist, eye, and mouth lesion. Instead, herpes simplex 2 infect below the waist. Genital lesions, because it's oral genital sex, can lead to HSV1 below and HSV2 above. So during oral genital sexual contact, virus can shift the place, but later it will come back into the specific place from the non-specific. What are the main clinical presentations of herpes simplex 1? Gingival stomatitis, keratoconjunctivitis, herpes labial, and temporal lobe encephalitis. On the picture, you can see how does virus replicate and inside a specific lesion and go out from it. Does anybody hear me?
Yes, doctor.
Okay.
Yes, doctor, we can hear you.
So very good. Let me a second to go back to the our presentation. Okay, can you see it?
Yes, doctor.
Yes, doctor, we can see the screen.
Okay, that's very good.
Yes, sir.
So, as I said, during the primary infection, the virus invites into into primary trigeminal nerve, and then can penetrate back after several kind of stimulation. It can back to the skin surface by these main three roads, main three ways, main three branches. So a primary infection, always infection of the skin and then penetration to the trigeminal nerve. And then during the latent stage, virus stay there and like sleep. And because it's under control of immune system. What kind of provocative stimulus can cause recurrent attack? First of all, that all the things which influence on the immune system and which can decrease immune status. I guess you remember that from the last semester, a part about boosting immune system, and it's I would say it's an part of non-specific prevention of recurrent attacks. So in pathobiological stage, I would say it's only human infection because it can infect only human cells and transmitted via saliva, and we can say it should be direct contact or indirect contact, but with liquid stuff. Virus invites mucous membranes and cause local primary infection. After that, typically asymptomatic, but can cause vesicular lesions that ulcerate in mouth or eyes, in this case. There is a picture of the child who has ocular herpes virus or herpetic keratitis. And you can see that recurrent lesions of herpes labialis, whether they occur on the near the lips or as you see on the picture the eye, and begin as tender itchy papules that progress to vesicles that burst brain and eventually scrub over. The source and fluids are highly infectious and should not to be touched. But with children, it's usually impossible. I mean, not touch it. So, diagnosis of herpes simplex 1 based on detection of the virus by PCR methods, especially for early detection in encephalitis for newborn baby. Multinucleate giant cells of tongue smear of skin lesions, eosinophilic Crowley intranuclear inclusions body on the skin biopsy can be used also and can be visible. So for treatment, primarily we use acyclovir or trifluridine, typically for topical for eye infections. Acyclovir penetrate inside this lesion and destroy the virus capsid. Moreover, it's stop spreading because it can cover it and became this lesion not so itch. So itching also limited by this treatment. Of course, acyclovir can be used like a gel for specific the place for topical, and also we can use this for systemic in pills primary for general infection. So about simplex virus 2 or herpes virus 2, as you see, it's totally same characteristic with herpes simplex 1. That's why it's because it's very similar, very related, I would say relatives. In pathobiology, again, only human are reservoir. Transmission by sexual contact primary. The virus invites mucous membranes, cause local primary infection, the same with herpes simplex 1, and typically asymptomatic, but can cause vesicular lesions in genital or perianal area. And I said if it's oral sexual contact, the primary infection can be seen nearby mouth, around the mouth, on the lip border. So it's a picture of the genital herpes of the man penis. Vesicle starts out separate, the same for herpes simplex 1, but become confluent and ulcerate into red erosions to tender that any contact can be highly painful. So this is infection, it's a painful infection, and for some cases, if it's only vesicle, sometimes a person is, especially if it's destroyed, can be missed, can be confused with other infections. But here it's usually not only one vesicle, and highly pain during the touching or itching. It's one of the important characteristic. If infected mother during the pregnancy didn't treat herpes simplex 2, during natural delivery, virus can contaminate newborn baby, which could cause herpes of the newborn. It's a very dangerous condition because we know herpes virus are neurotropic virus, so infect nervous tissue. And immune system of newborn baby are not able to fight with infection. So baby can be born with such lesions, or lesions could develop them to 1 to 2 weeks after birth. Detection. Totally same detection, but only difference here we will take from another part. So anyway, in both cases, infected part. Treatment, acyclovir is totally same because the virus is similar, very similar, and acyclovir also effective against simplex 2. So prevention here for newborn baby, for neonatal herpes, is a cesarean section in infected mother. Will prevent because the virus not penetrate through the placenta into infant. Only contamination happens only during natural delivery. So that's it about the herpes viruses. About other viruses you can read by yourself in the books which I sent to you, and it's a pretty nice described everything. And I would like to tell you about prions, few words about prions, because this topic usually skipped because it's the last, and I will tell you why it's not very popular topic. So what is it, prions? Prions are group of non-cellular infectious agents that are not viruses and really belong category all by themselves. The term prion is derived from the words proteinecous infectious particles, because it's just two parts, and it's naked protein molecule. Till the 1918s, it was unknown viruses, but the Prusner, who described the prions, got a Nobel Prize for his work, for the prion developed. It's not only human infection, it's a primary animal infection, and it was described in animal, but humans are also host from similar chronic diseases, including Creutzfeldt-Jacobs diseases, Kuru, fetal familiar insomnia, and some others. Here you can see the main differences between prions and viruses. Look here, virus. Usually it's filtrable infectious agents, both of them. What does it mean, filtrable infectious agents? Does anybody know? Filtrable infectious agents. Nobody know. What does it mean, filtrable infectious agents?
They can pass through any barriers easily.
You are close, Ruti. Not any bacterial filter. So these are small microorganisms, but the prions is not organisms also. Anyway, very small, so can pass through the bacterial filter with very thin pores. Okay. In prions, there is no nucleic acids. There is no morphology which can be described by electron microscopy, because it's just proteins, nothing more, proteins without DNA or RNA or capsid or cell wall membranes, nothing, only protein structure. Of course, it has proteins. Look here on this part. It can be disinfected by formaldehydes. Viruses could be disinfected, but prions not. Proteases enzymes. For some viruses it works, so some viruses can be killed by protease enzymes, but prions not. Heat up to 80 degrees, so we suggest mild boiling will kill most of the viruses, most of them, not all, but most of them, but not kill prions at all. Ionizing and ultraviolet radiation also can kill viruses, very effective, but prions not affected. So what kind of disease? Actually, not disease exactly, but what kind of effects it could cause. Cytopathologic effects, when the virus, for example, change a cell. It could observe in the viral infection, but not observe in the prion. Incubation period. I'm sorry, wait a second, please. Sorry. Incubation period for all prions, it's very long incubation period, but for viruses it depends on virus. We know for some viruses it's very short incubation period, like influenza virus, for example, couple of days, but for some could be long, for example, for Herpes virus, several months usually, but for prions it's usually very long, more than several years. So for the viral infection, there is no any kind immune response totally. So there is no even cellular or humoral immune response. Also, there is no interferon production to it, and there is no inflammatory response. So our immune system totally disabled here. So how does prion infection looks like. On the cellular level, it can be seen under the microscope after the biopsy of nerves. And on the picture, you can observe prion proteins fibers are red. It's when the prions are placed in the cytoplasm of the nerve cells here. And as you see, there are many proteins, not our proteins, but prions inside the nerve. What does prions do? And on this diagram, you can observe what's exactly happens. And I will read you, but you can read it by yourself. On the first step, it's here. The prion protein infect a nerve cell and penetrate inside. So the pink one, it's prion proteins. Gray, it's normal proteins of our own cell. Look what's happened on the second stage. It's one of the primary stage to understand what's exactly happened. Prion and our protein should be in contact. So it should has direct contact, and the prions are able to shift the configuration of the normal proteins, converting them to the prion. Of course, not all proteins will be converted. Only some proteins which similar by the structure to the prions will be converted, and all of it which in contact will be converted. Then when the conversion process is finished, large numbers of the prions bind tightly together and form elongated chains. You can see elongated chains here of the proteins. So it's not separated right now, it's bind in chain. And then when the chains build up, they form fibers within the cell which interfere with the host cell function and destroy it. But not exactly destroy, not disrupt, but destroy the function of nerve cells, primary. And destruction is not disrupting, it's destruction of the cell function and sometimes connections between cells. Oh, that's it about prions and herpes viruses. Tell me, please, do you have any questions about herpes simplex, herpes viruses and prions?
Yes, I have question.
No,
Does it replicate as it doesn't have an DNA or something to replication, just only protein?
Okay, very good question. Look, it's not replicate, and you can see on the picture here. And as you see, there is no replication process, but it's like, how to say, did you watch any movies with zombie? Zombie not replicate, but increase its numerous, but by infecting other normal people. And here is a similar process that prions will change our normal protein, and normal protein became prions. Is it clear now, more or less?
Yes.
Okay, good.
Yeah. Thank you.
Anything else?
Sir, what can be the treatment if the person is infected with prions?
So Yatri, there is no any treatment strategy. So prion infection, untreatable infection, totally. If a person get prion infection, means it's cause death. But fortunately, it is very rare infection. There is not many cases over the world, less than 1,000 cases per year about prion infection, and it's very long infection, I would say a very long incubation period after getting the disease, and then very long period of how to say primary symptoms until the acute stage. So a person could live with prion infection more than 10, sometimes 30 years.
So
Okay,
doctor,
sir.
it has the same case as like HIV?
No, no, it's absolutely independent diseases, because first of all, HIV, it's virus. Here it's just proteins. Second thing, absolutely different transmission ways. For HIV, you know, it's a sexual contact, blood transfusion, organ transplantation, and
Yeah, I understand. I'm talking about the part where like someone can be like infected with it, but doesn't know like if they have it or not. Like that's what I'm talking about.
yeah, let me finish with the transmission way, because I didn't told, I guess, about how does prion infection transmitted. So you can get this infection with food primary, to eat primary meat, infected meat, under cooking. As you remember, prions not killed under the heating, mild heating, and especially for meat eaters. So person who would like to eat a lot of meat, and of course like steaks with not well cooked but raw, and it's a main way here about the transmission way. And about your question to understand how it's what's also, it's also different. But look, because for HIV right now we have, I would say in Russia, we have a very nice opportunity. You can be checked for HIV for a couple of days without any kind of borders and absolutely free in all cities. In all big cities, so we have a lab who can take this analysis and will give you result. Moreover, you can buy in pharmaceutical store like express test, which will give you result less than half hour. So for prion infection, such kind of diagnostic methods, it's not available. That's why it's more problematic in this case. But HIV is very contagious and transmittable disease, because you can't share prion infection with some other people, instead HIV. So if person infected, it can be a source of infection. For prions, infected person is not a source of infection, only if someone will eat infected person. Anything else?
Understood. Thank you, doctor.
You're welcome. So if there is no any questions, please right now type your name in the main chat. Okay? Please type your name.
Yes, sir.
Yes,
Yeah.
sir.
Yes.
Yes, sir.
So I will understand you listening here, and we will continue 5 minutes later. So let's continue. The second part of our today lecture will be about everything. I tried to give you the most, I guess, useful information, perhaps, from all the topics what we had in the primary. We'll talk about some definitions, and as I understood, the main unclear things. So first of all, here about our normal microorganisms and abnormal microorganisms. So normal flora is the organisms is always on us or inside us, in gastrointestinal tract, genitally, urinary tract, and oral mucosa on the skin, you know where. So, and as you remember, there is some sterile areas of the body. It should be blood, cerebrospinal fluids, so organs, and like liver, heart, you know. And if we found any microorganism here, it indicates something is going wrong. The first indicators is described glucose increased proteins and increased proteins. So opportunistic pathogens, it's also normal flora organisms that becomes a pathogen when the host immune system defense decreases, or it will come into non-specific areas from, for example, from surface of the skin. It comes inside the skin during some trauma, surgery, operations. From urinary tract mucosa, it can penetrate deeper in during catheterization, for example, or immune system is fall down after the chemotherapy or some other infections. It's one of the primary ways. So I guess it should be clear. So during this part, during overview, please, if you will have any questions, just interrupt me and ask, because I'm talking about absolutely different things here. And if you have any questions, please ask. So next part about tissue tropism. It's when the organism are selectively can infect a particular tissue or an cell. An organ. For example, influenza virus does not affect your hand. Gonorrhea only infect mucosa. So such kind of tropisms can be species organ or non-specific. So what has been organ tropism? We know about hepatitis viruses, which primarily can infect only hepatocytes, or some non-specific tropism. It's for Staphylococcus aureus, which can infect a lot of tissues. So HIV integrates a provirus into our chromosome, and antibodies actually assist the HIV organism because opsonization helps to internalize the virus. Because of almost no cell-mediated immunity, so T cells are infected already. 8 patients die from TB, cytomegalovirus, actually some candidal infections or pneumocystal infections, which fungal infections. When a person has a second infection, it's called co-infection. So in this case, HIV virus has specific tropism to immunocompetent cell, but AIDS, it's not a particular disease, you know, it's a complex of syndromes. So we also have obligate pathogens instead normal microflora and opportunistic pathogens. Obligate pathogens will always cause infection and disease whenever whatever they are encountered. For example, Yersinia pestis cause plagues. For example, Bacillus anthrax cause anthrax, and the smallpox virus cause smallpox. Always, and doesn't matter how does our immune system will influence on the process, such microorganisms always cause the disease. However, they don't want to kill their host. It's a problem for microorganisms and for us, of course, because if the more adapted virus or bacteria, it's not harmful but very replicatable, like herpes virus, what we discussed today. Very educated, very nice virus because never kill us, always with us, and fast transmitted virus. So this virus really won the competition to get a very huge amount in our population. So, you know, more than 90% of all humans infected with herpes simplex virus. So instead, other viruses which can kill us, very limited, like smallpox virus, you know, it's limited by vaccination, but I guess if this virus wouldn't cause so dangerous disease like smallpox, but cause mild infection with some lesions, for example, as herpes virus, nobody will make such vaccination, such strong program of vaccination over the world to limit this disease. So next part about virulence. So virulence, you know, it's ability to cause infection and disease. It's like not exactly an ability, it's also a measurement of pathogenicity. Multiplicity of infection, inoculum, is the number of organisms needed to cause disease. There is an inverse relationship between virulence and multiplicity of infection. If only a few bacterial cells cause the disease, it's said to be very virulent. We're told for Escherichia coli, for example, it's a very high infection dose, so inoculum. But for Shigella, which is very similar, very low infection dose. Obligate pathogens are virulent when with a low inoculum because they are effective. Most inoculum needs 10 raised 3 to 5 organisms. Only 10 organisms can form an inoculum in a virulent organism such as anthrax. So for the anthrax, as you see, just 10 microorganisms, so 10 spores or bacteria in inoculum can cause anthrax. Anthrax avoids immune defense by exploiting the very mechanisms used to fight it. It becomes activated after phagocytosis. So anthrax spores penetrate inside human body and do nothing. Then phagocytes eat the spores, and at the same time, the spores became active. Another bacteria, Mycobacterium, which causes tuberculosis, will coat the wall of a vacuoles, so lysosomes can't fuse. So again, escaped under phagocytosis. Rickettsia, or very small bacteria, as you know, it's on a timer and escapes the vacuole to invade the nucleus. So Rickettsia, as you know, it's obligate intracellular parasites. Different relationships are possible between us and microorganisms. First, very known for us, infection. When microorganism present in our body and cause infection. So another condition, disease, we would say, when adverse symptoms due to infection is visible. So infection and disease, it's different things, because infection, it's just a presence of microorganism. But sometimes it's not a disease, especially if it's not danger for us. So about normal flora I told about pathogen also I told. Immunocompromised, you remember, it's people who has suppressed immune system or deficient immunity. Definition from a first lesson from the last semester. Nosocomial. Nosocomial infection, that's infection acquired in hospital setting. Very dangerous for a particular person, because usually microorganisms which cause nosocomial infection are multi-resistant to antibiotics. Community acquired means acquired in routine day-to-day activities. So it's usually for people who are working in the same area and do the same job. Opportunistic pathogens, I told about normal flora from plus immunocompromised means pathogenic microorganism in main cases. Obligate pathogens always cause disease if present. Pathogenesis, it's events leading to disease. Pathogenesis you also would understand as traveling of the microorganism inside a human body, some human body. Multiplicity of infection, it means inoculum required for infection. Incubation period, it's the time from transmission to first clinical manifestations, to disease, as you know. Virulence, it's a measure of disease capability. So virulence, it's a measure of pathogenicity. We know we can measure virulence because of measuring virulent factors. So virulent factors here is usually ultrastructure or exoenzymes or exotoxins. Bacteremia, when bacteria are present in bloodstream. You know, abnormal process because blood should be sterile. Bacteriuria, when bacteria are in urine. So here it's a very big like trick, because bacteria should present in urine in small amount, but usually it's normal microorganisms which present in small amount. If pathogenic microflora present, of course we call it bacteriuria, or normal microflora present in very big amount there. So pyremia, when gram-positive bacteria cause bacteremia, and means gram-positive bacteria present in the blood, and it causes usually very high fever. Pyrogenic, it's a fever-producing infection or bacterial components which increase not only immune response to get body temperature higher, but also release some components which can destroy cells inside blood or mucosa. Purulent, it's a pus producing, rich and open to drain out. The pus is a collection of dead neutrophils, which are the first immune cells that are present in the pus. Granulomatose, it's an infection where macrophages are first immune cell, but not pus. Differential diagnosis at the all possible cause of diseases when a person usually just guess, perhaps like a case study, perhaps it's a one causative agent cause the same symptoms, but perhaps another one because it has absolutely similar significant effects, or it can be transmitted by the same way. That's why it should be same or similar. Urosepsis, it's a septicemia from urinary tract infection. Facultative intracellular parasites, it's not obligate pathogens. They can survive temporarily in white cells, such neutrophils, monocytes, and macrophages, like we told about bacteria with capsule around it, or spores. So exotoxins, toxins made and then secreted out of the bacterial cell. Endotoxin, it's a component of the cell wall which is toxic. It's a part of lipopolysaccharide of the plasma membrane of gram-negative bacteria only. So host defense, as you see here, host defense, host defense we can divide into non-specific defense, which first line and second line, and specific defense, which third line of defense. So first line, it's usually skin, mucous membranes, or some chemicals which released on the skin. It's very nice barrier because unpenetratable for most of microorganisms. So if some injury or abnormalities, microorganism could penetrate inside. Here, second line of defense could stop the infection. For example, phagocytosis, complement parts of, you remember, it's a proteins, interferons, some inflammation process, even and fever could stop the infection, and it will be the first line. Third, and we would say it's specific, lymphocytes, which also could produce antibodies. It's the specific line of defense, because antibodies, it's only structure which can specifically bind an antigen to destroy or remove whole pathogen out of the body. You remember, we called T helper cells CD cells. Once again, why we call it CD? Because it has specific cluster of differentiation on its own surface. So each T cell has its own cluster of differentiation on the surface, and right now here you can see how it looks like on T cell or B cell. So such cluster of differentiation, it's of course it's in summary, because such clusters not always present in such big amount here, but only as a picture to show you all possible main classes or clusters of differentiation. So let's a bit talk about epidemiological cases. So reservoir of infection could be different. Very rare, it's we are. So for some pathogens, we are can be a source of infection, but it's in particular cases, primary pathogen should come outside. So there are three main types of reservoirs. First, animal reservoir, second, human, and third, non-living reservoir. It's like a soil, water, air, like this. Animal reservoirs we also can divide into zoonosis, when disease naturally spread from animal host to human. The main example here, rabies. Another one, acquired zoonotic through various roads. Could be direct contact with animal and its way, eating animal, blood-sucking arthropods. So zoonosis can be real zoonosis or anthropozoonosis, because humans are usually end host of zoonotic pathogens. So bacteria or virus will die inside us or will die with us. Human carriers. You know, a lot of infections spread from people to people, and some individuals eventually develop illness while others never get sick. Very nice example here. It will be hepatitis A virus, when a lot of people not allow any kind of clinical manifestations. Instead, other people very suffer from it. So also healthy carrier may be a defensive system that protect them, perhaps. So non-living reservoir, as I said, soil, water, food can be reservoir of infection. But what's the main problem here? It's to understand where from bacteria come into soil, water, air, and food. If is the source, specific source, like a human, for example, which sick, we would say it's only transmission way. But if food by itself can be a source of microorganisms in small amount, but then during the storage condition, microorganism increase it amount or increase something, so I mean produce toxins in the food, we would say about the problematic non-living resource here. Of course, you know, in soil there are many pathogens, leaves, and most of them comes from animals or humans sometimes. And water is the source, and also because of contamination, because bacteria or viruses come from the soil or from infected people. So the nature of infection disease, we have to make clear some definitions. So what the symptoms? Symptoms is subjective characteristic of disease felt only by the patient. For example, pain. Okay, you would say we can measure a pain, but of course it's better to ask, how do you feel yourself? What kind of pain? Where is it? Things, it's objective manifestations of disease observed or measured by others. For example, high fever, we can measure it, or rash, we also could measure it, or for example, muscle contraction, we also can observe it. So syndrome, it's the symptoms and signs that characterize a disease or abnormal condition. So there are several different results. Asymptomatic, when a person infected but have no any kind of syndromes. Sometimes it's subclinical infection, when it's uncommon, not very clear, non-specific symptoms or things. And we would say, I would say full clinical, when a person feel everything what written in the book. If rash, it's rash present, fever, back pain, etc. And in the end of the overview, I would like to tell you about this scam of immunity, which we discussed last semester, and right now we have to refresh it in your memory. This scam will characterize a type of immunity after injection, injection of the vaccine or getting the disease. And also it's just describe the types of immunity, all types which possible in any cases. So we know immunity can be innate and acquired. So innate immunity, we know that our innate genetical difference which make us non-sensitive to many infections which important for animals or plants or bacteria even. For example, you never could get plant infection because we are different. So it's kind of innate immunity. Then acquired immunity. Acquired immunity, that kind of immunity which we get during the life. So we can get it naturally or artificial. Naturally, how we can get natural acquired immunity? During active, if it's active means we got an infection and our immune system solve the problem and produce antibodies. And we have immune memory which stay with us all the life. So if, for example, or not all the life anyway, during short time sometimes, okay, good example here be influenza infection. A person get better and his immune memory cells are produce immune memory and protection. In this case, we will talk about active, natural, acquired immunity to influenza virus. Passive natural immunity, possible only in two ways, both related with mother and newborn babies. What's exactly? Mother's immunoglobulins, IgG, could penetrate through the placenta into fetus, and fetus are protected by mother's antibodies. It's passive natural acquired immunity. Moreover, after birth, if mother use breast milk for feeding, infant can be protected by the same IgG, which also penetrate into mother's milk and then into newborn baby body. It's also passive natural acquired immunity. So baby can be protected from influenza by mother's immunoglobulins, by mother's antibodies during some time. It's passive because it's artificial, but not mother's, not self, but mother's antibodies. So about artificial acquired immunity, it also can be active or passive. Active only after vaccination. So this one, active, only after vaccination. So if a person gets a vaccine, his immunity will form active artificial acquired immunity to influenza virus. But also it can be passive if we will use serum for treatment or for prevention the infection. So if the person get artificially made antibodies, or to treat infection or to prevent the infection, it will be passive artificial acquired immunity. So all these natural and artificial active and passive immunity can be monovalent against single antigen. Very good example here, infection of influenza again. If a person was infected, he has acquired natural, active, monovalent antiviral immunity. If a person get a vaccine include several strains against microorganisms, against influenza. So vaccine against influenza virus are trivalent vaccine. So here we talk about acquired, artificial, active, polyvalent antiviral immunity. So if person get a vaccine, as you see difference here in artificial and natural, and how many strains. Of course, if we will use a vaccine which include only one single antigen, it will be acquired artificial active monovalent antiviral immunity. So the same way we can use to characterize all kind of immune response against a pathogen or immune response after getting the vaccination. And also we can characterize passive immunity if we talk about serum injection. So that's it. I am open to answer for your questions. Anyway, if you have any questions,